Okay. Thank you, Eric, and it's really been amazing to be here at the ISB and hear the seminars and to see this amazing place. And so I am not going to talk about the skin microbiome today. I'm going to talk about the system that I've been thinking more and more about, which is hospitals. And, you know, the old adage of if you want to stay healthy, one of the best things you can do is stay out of the hospital, especially if you're sick. So I'm going to talk about tracking hospital infections. This is something that I think the system is actually quite complicated in terms of what is your exposure to multi-drug resistant bacteria. So that I don't forget, a lot of the work that I'm presenting today is from Evan Snitkin, who's a postdoctoral fellow in my lab. But really the way that this work is possible is by being at the NIH in a very integrated group of bioinformatics, clinical epidemiology, hospital infection control, and clinical microbiology. So this is Tara and Patrick. And, you know, we have to work together as a team. So the system is all of us. So it's really this interaction between genomics, bioinformatics, infection control, and clinical micro. So the issue that I'm thinking about is the gram-negative bacterial infections and the fact that antibiotic resistance is really on the increase amongst all of these. So, you know, what we're seeing is that there's this, you know, a large fraction of the ICU infections are caused by these different gram-negative bacteria, and we're talking about a significant amount of infection and mortality, as you'll see in my slides. Pseudomonas aeruginosa is really important. I'm going to talk about Klebsiella and about Acinetobacter today. And you know, one of the reasons that I worry about these is, first of all, we've had outbreaks, and second of all, because the rate of multi-drug resistance is really increasing amongst these. And so what you have to understand is that we have run out of drugs. You know, there's the first tier antibiotics, second tier, and third tier, and then we just don't have anything else. So, you know, I started getting involved in this when, you know, it became clear that what we have is palliative care. Okay, so this was our 2007 outbreak of Acinetobacter baumannii at the NIH Clinical Center, in which 30 patients were infected, and 40 patients were infected, and 10 of them died. And an outbreak in a hospital, so I have to tell you, this is always going on. What I mean by an outbreak is when in the clinical center you start seeing an increased number of patients who have these multi-drug resistant bacterial infections, and you start to see that it is spreading patient to patient. Now, I'll come back to that at the end, because I don't mean patient to patient like the two patients are hanging out playing video games together. That's part of the system, is how does it get transmitted from one patient to another. But in this case, what we had was we always have a low level of Acinetobacter baumannii multi-drug resistant Acinetobacter baumannii. But in this case, what we see is that, you know, oftentimes we'll have 1 or 2 cases, but then we started to get these, you know, 8 to 10 cases. And to actually finally rid ourselves of this infection, we had to close half the ICU, not, you know, and basically clean the entire thing and not allow any existing patient in, and then close the other half of the ICU and do that same thing. What we had here that we couldn't understand is, in the context of this outbreak, we had 3 isolates that were simultaneously in the hospital. And that's one of the funny things about Acinetobacter infections, is that when they strike, they often seem polyclonal. And so, you know, from a genomics perspective, what we're wondering is, how does this happen? Like, we have these 3 similar strains, and what we wanted to know was, you know, is there an ancestor? And, you know, is there 3, did that derive, did 3 strains diverge from it, or did we have 3 separate colonization events that happened in the hospital? So we sequenced the 3 isolates, and actually it was very confusing to us until we assembled the genomes. And when we assembled the genomes, what we could see here, these are our 3 strains that were in the hospital, strains A, B, and C. And I've I've seen this, this is Circos, the Mark O'Mara drawing program that is used a lot for the cancer genome, but I think it's really fabulous for microbial genomes that actually are circular. So aligned relative to an ICU strain from Rome, any time that the sequence is the same, it's white. But if it's a SNP between ICU and A, it's shown as blue. And if A and B are the same, then B is blue. But if B is different than A, it's red. And if C is different than if C is different than A and B, it's shown as green. Whereas when it's red, it means that it's the same as B, and when it's blue, it's the same as A, but different from ACICU. So what we could actually see was that there are a lot of SNPs, and that's why we couldn't build a genetic map originally just by using all the SNPs, because in fact these SNPs are not independent of each other. This is a huge block of recombination that's occurred. So we only use the SNPs that are outside the blocks of recombination to build the genetic map. And in fact, at that point, what we could understand was that ICU and A are as different from each other, but B and C do appear to share a common ancestor. So actually A is different, but B and C shared an ancestry. Now, you say this, and then when we say this to the clinical micro and the hospital infection control, oh yeah, actually B and C were first isolated from the same patient at the same time. So, but what I'm saying to you is that this is a very complex system, and it all seems like there's many possibilities. And when you use genomic information to come in and say, this is what I really want you to tell me, then there actually often is that epidemiologic evidence, but you have to go at it in a directed fashion. So what this would suggest is that how could that be possible that B and C would have come in together? So that's when we started looking at what were these areas of recombination. Again, this is the Circos drawing program. And what you can see here is that this is a zoom in of the region that was right at the top of the screen near the origin of replication. And what you're seeing here is that these ribbons, A is very similar to B and C, but this area has changed completely. What has happened here is a wholesale swapping out of the O antigen biosynthetic locus. I'm talking about all new genes coming in. So the O antigen biosynthetic locus is part of what the immune system is recognizing. So the patient who was colonized with B, then even though that patient may have started to mount an immune response, can still be colonized with strain C, because this now goes stealth below the immune system. And so this is how we could now imagine that a patient would come in subclinically colonized with 2 isolates and could start to spread that in the hospital. So B and C, we now understand they came in together with the same patient. And what about A? Well, A was actually the first isolate that we picked up. But now we had these tags for A because what we had was the O antigen biosynthetic locus that was unique to A. And we made out PCR amplicons from that region and screened through all of our non-drug resistant Acinetobacter baumannii and found the single one that existed in the hospital a year and a half earlier that didn't give any, there was nothing about it except that it had that PCR amplicon. So we sequenced that isolate, HC64, and in fact what you can see is that it is nearly identical to strain A, except that what is not shown here is that now A has an enormous insertion. It's kind of compressed here. A has an enormous insertion that brings in a multi-drug resistant locus. So what we see from this is that a non-drug resistant Acinetobacter evolved drug resistance within our hospital, which means that we also need to start thinking about better stewardship of non-multi-drug resistant isolates that are in our hospital. And so we now, we have this situation. It happens, it happens every month in our hospital. We are always wondering, are we about to start an outbreak? How early can we detect that? And so we always have these Acinetobacter baumannii. And the question is, how are we going to quickly figure out when we get, a new case, whether this is hospital transmission or whether this is a new isolate that's come into the hospital, because that actually triggers two different decision processes. So we've been looking at optical maps, which you string out the DNA on a slide and you actually do the restriction digestion in silico, and that gives you a really nice resolution. That is digital information that can be shared between hospitals, because we need to start increasing. When we had this outbreak, we were wondering, do we have the same strain isolate, in our hospital as at Walter Reed? We couldn't figure that out. This is the type of information we could share. But one of the things that I think we also have to rethink in terms of these diagnostics is that we now have whole genome sequence data and we're not using it. So we've been working in the lab now to make predictions of what regions really should be used to test for generating PCR amplicons to say, do we have a new isolate, or do we have hospital-acquired transmission, and to really redo the process of clinical diagnostics based on whole genome sequencing data. So we've looking at all of 26 of the sequenced Acinetobacters from all over, we've been designing PCR primers that now can give unique signatures with just a few steps to say, what isolate is this? And they're actually just more, they're more sensitive than any of the existing means that are used, like multi-locus sequence typing that often can't, in all of these cases, this means that these two strains can be distinguished by two different PCR amplicons that we could design, but they're not distinguished by multi-locus sequence typing. Okay, so that's sort of prospective what we kind of went through in dealing with the Acinetobacter infection. So now I'm going to talk about Klebsiella pneumonia, which is really another emerging pathogen, particularly in the Northeast U.S. And so, when you think about where you're going to a hospital for a transplant, for chemo, for immune, for some sort of major procedure, especially it's going to make you immune compromised, most people think about where's the best clinical team, and one of the things that you actually should be thinking about, especially if you're going to be immune compromised, is who's in the middle of an outbreak. Unfortunately, you can't get that information. When I said to the head of hospital infection control, like, what would I do if my father needed, to undergo, some sort of stem cell transplant? And he said, I hope you would call me. I can get that information for you. But we really need to start, making hospitals accountable and making that information more available, because that really is one of the things that you have to start thinking about. And in terms of that, if you even read this week's New Yorker, and they're talking about medical tourism, so your graduate students sort of showed it. One of the things that I do worry about is people going abroad for medical treatment and bringing back these infections, because, that really will delay your recovery. Okay, so I'm talking too long. Let me talk to you about KPC. So Klebsiella pneumonia, this carbapenemase-resistant Klebsiella pneumonia, this is incredibly scary stuff. This is just, this is pervasive in ICUs, particularly in New York. So what happened to us? So this is all really new data, as you can see, because this was all being rolled out this fall. We had a patient come into the hospital. We knew that she was KPC positive, so she was seen in isolation. And we felt really good about ourselves as a hospital because she was released. We did these throat and groin cultures, and for 3 weeks, nothing happened. And we sort of started thinking, well, we really learned from the Acinetobacter outbreak. We really know how to keep these things under control. So she was released on July 1st. Or sorry, maybe July 15th. But then in August, we started getting patient 2, 3, 4, 5. So sorry, when there's a black line, that's when the person cultured positive. When they're in blue, that means that they're in the ICU. In gray is someplace else in the hospital. And in red, we ended up, as you can see on this day, creating a cohorted area where all of the medical care for those people, that person could only serve the cohorted patients. That person could not then do anything else in the hospital. Because what ended up happening was that 17 patients were infected and 10 of them died. And so the first question is, this person came in KPC positive, but then was released. So are these other cases even related to patient number 1? Or are patient 2 and 3, like, did someone else bring in a new isolate and that's what's circulating around our hospital? Did we, is this just coincidental because this was, patient 1 was our first KPC in the hospital? Maybe it's just, it really is increasing like that. So we sequenced all of these this is just to scare you about the kind of thing that goes on in hospitals. So, and we really do call it the attack rate. If you were in our hospital in the ICU for 2 nights, you had a 1 in 6 chance of getting KPC infection. But if you were there for 4 nights, you had a 1 in 3 chance. And that basically held for 4 months while we were trying to figure out what the hell was going on. Okay. So if you didn't have genomic data, if what you had was the system biology, you're going to incorporate all the information. This is even just like who overlapped in the ICU. I'm not even showing you the maps that we drew, which were like who shared a wound care nurse, who shared a physician team, who shared a ventilator, who shared a bed, who shared a this, who shared a that, because there's a lot of scenarios that we can draw up, and this is even just, who physically overlapped in the ICU. And it's a mess, and we can't figure out what are the at-risk procedures. So we sequenced all 18 of the isolates, and what I'm showing you here are the SNPs. So this is a 6 megabase genome, and basically there are 40 variants. So what I'm already telling you, I would prefer that you not take pictures. I mean, if that's the procedure here, that's fine. But otherwise, I'm giving a talk. But if that's the procedure here, I'm willing to live with that. So in this, we have 6 million base pairs, and we have and so I should say, if you want me to send you the data, I will, and you can tell me what you're going to use it for. So we have 6 million base pairs, and there are 40 variants. And so first of all, what we can clearly see is that patient 1 is part of this outbreak. There are these 3 SNPs that are shared amongst these isolates, and then there are these okay, I'm getting ahead of myself. But basically, what you have is a 6 million base pair genome, and you have at most 40 variants. So even though, even though these things go through bottlenecks, patient number 1 is related to the whole outbreak. So how is that true? Well, what we see is that from patient number 1, there are 2 different transmission events. One that leads to this yellow cluster here, and one that leads to this green cluster here. And in fact, these 3 SNPs are shared with this cluster, and what we really saw was that patient number 1 had a mixed infection, where in some parts of that person's body there were these 3 SNPs, and in other parts there were not. So But what you see is that means that there's definitely two separate transmission events, one from patient one, to this cluster and one to this cluster. And then those people take off. Now, also, in terms of, what are the risk factors, because I'll dive more into the scenario of the outbreak. What were the risk factors that we saw? Well, we definitely saw, as an example, this was a ventilator that had been used on patient number 6, had been cleaned extensively, and then we cultured KPC out of it, and it clearly matched patient 6. What we actually did was to try to think about using this genomic data. If you just use the genomic data, this is the map that you would draw. And it's a little bit unsatisfying because you really still have kind of this broad range idea of what could have happened. But if you incorporate now epidemiologic data with genetic data, you start to actually get a very fine sense of who transmitted to whom. And it's incorporating that sort of thing of who was in the ICU at the same time, who had the same wound care nurse, but in our algorithms for doing this, the things that we consider are genomic data trumps everything. If it's not consistent with the genomic data, I'm not interested in looking at it any further. But once you have the genomic data, we use as tiebreakers that there are people who are silently colonized. And that's what you actually had to intuit by the fact that the patient was left the hospital July 15th, and we didn't get our next patient until August 7th. What that means is that those people have, even though they're immune compromised, that they can spend long periods of time in which they are silently colonized. And by that, I mean that we are not detecting that they are colonized. And so within the algorithms, genomic data trumps everything, and then we count the number of days that the person would have to be silently colonized, going back to when, for example, patient 4 and patient 10 overlapped in the ICU. How would you minimize the number of days that those patients had to be silently colonized? And this kind of data is actually really just hypothesis generating information. What we're doing now is that we use this to then work with hospital infection control and say, okay, here's what we think might be the at-risk procedures. So for example, if patients 6 and 14, if I feel that there's a strong link between 6 and 14, tell me what they do have in common. They don't have anything in common. Patient 6 left the ICU before patient 14 came in. 2 days before patient 14 came in. Okay, what room did patient 14 go into? Oh yeah, the same room that 6 came out of, but we cleaned it. Okay, so, what we ended up doing that really ended up stemming this infection was, one of the things was that we found KPC in the sink drains. We found it in the sink drains of 6 ICU rooms. So someone comes in, they wash their hands, that's great, but it splashes back out of the sink. So patient 1 to patient 4 is an interesting case because we have no epidemiologic link between patient 1 and patient 4. We could not find that they had anything that would put that at risk. That's a really interesting case for us. So what we did was that we went and looked. We took all the patient trace data for the entire hospital, and we said, tell me who overlapped with patient 1 after, patient 1 became infected. Oh, I'm sorry, patient 1 always came in infected. And then overlapped with patient 4 before patient 4 became infected. And amazingly, of the 300 people that, are in our hospital at any one day, there are exactly 5 people who overlapped with patient 1 and then overlapped with patient 4. So now I'm showing you a more colorful version of all of the wards on our hospital. And what you can see, again, the black line is when patient 4 came up positive, and that these 5 people all had the possibility to have been the true silent colonized individual. That is to say, someone who was infected and never developed an overt bloodstream or urinary tract infection, but was actually passing it on. Now, one of the things to see here is that patient 4 came up positive right here at the end of August. All 5 of these patients are still in our hospital, and we didn't, so this is where we need to speed this up, because now these 5 patients are patients that need to be undergoing additional surveillance. You just can't have them out in the general hospital population if one of the 5 of them may really be silently colonized. So I guess this is going at the point where these are the kinds of things where I think genomic information and the fast pace by which we can do genomic sequencing is really going to change the practice of medicine, because we're not devising new antibiotics. What we need to do is to really get better at not exposing people. So when we use epidemiologic data, we need to capture the probabilities of the transfer based upon the donor and the recipient status. And that's what I think we need to even go further out on, is to try to understand what are the medical procedures or the underlying baseline data that would make it so that someone would be likely to become infected, to transmit. We really also need to start to incorporate those systems biology of what's the, of the role of hospital personnel, of equipment, of all sorts of at-risk procedures, and to get out in front on tracking nosocomial transmission routes and finding the weaknesses in our hospital. So I'd just like to close by acknowledging the people in my lab who are amazing, in particular Evan Snicken, who is the bioinformatics person doing these experiments, the NIH Sequencing Center, that when I tell them, I really need this data and I need it, in 2 days, they actually don't hang up the phone. Department of Epidemiology, because what's really important here is all the clinical metadata that we have underlying this, and the Department of Laboratory Medicine. Thank you.